cDNA libraries. cDNA library is a combination of cloned cDNA fragments that are inserted into a suitable host organism or cells. As I told you that cDNA library, it is constructed from messenger RNA by the process of reverse transcription, where messenger RNA it is converted into cDNA. So genomic DNA it is not involved directly in cDNA library construction. So for this purpose, messenger RNA it is extracted from a specific cell types where all the population it is expressed, and then it is extracted and converted in vitro in cDNA components or fragments, and then later on they can be used for cloning. So we can say that cDNA library it can only represent a one small subset or component of the you can say that of the whole genome of a particular organism. The reason is that as compared to genomic library, the components of messenger RNA they may be belong to specific tissue type, and they can expressed variably. Some tissue may have high expression of a particular messenger RNA as compared to other. So it can represent only a small subset of the total genome of an organism. मिसाल के तौर पर अगर हम कंपेयर करें किसी हायर ऑर्गेनिज्म के किसी कंपोनेंट को या पार्ट को, लाइक प्लांट रूट्स को, तो प्लांट रूट के मैसेंजर आरएनए से जो लाइब्रेरी कंस्ट्रक्ट होगी वो डिफरेंट होगी उस cDNA लाइब्रेरी से जो कि लीफ से या पत्तों से कंस्ट्रक्ट की गई है, क्योंकि दोनों ऑर्गन्स में, रूट में और लीफ में, जो मैसेंजर आरएनए का एक्सप्रेशन है ये डिफरेंट होगा। और मैसेंजर आरएनए का जो एक्सप्रेशन है ये ऑर्गन की जो मैच्योरिटी है या लाइफ है उसके साथ साथ भी ये वेरी करता रहता है। सो इसलिए ये कहा जाता है कि जो cDNA है ये रिप्रेजेंट करती है एक स्मॉल पोर्शन ऑफ जीनोमिक DNA, not the whole genome. So then we can differentiate between cDNA library and the genomic DNA library. When we have a look on the cDNA library, we will note that it can only clone those bases that are present in the messenger RNA, not the whole gene, because a portion of the gene it is converted into the messenger RNA. So cDNA library it represents those nucleotide bases that are present in the messenger RNA rather than in the gene. On the other hand, genomic DNA library it can clone the whole gene fragment. The second difference is that the messenger RNA in case of cDNA library its expression may be variable. For example, in some tissue the expression may be high, so the particular gene may be present in high number, whereas in some tissue the expression is low, so other population may be present in low number. So those that are enriched they are present, they can represent cDNA library more frequently as compared to those fragments that are expressed rarely. But on the other hand, in case of genomic DNA library, it can represent the whole complement of the genome or the chromosome. So it is not based on the expression as compared to cDNA library. When we note the expression of a particular gene, so it may be high abundant, or it may be moderately abundant, or the expression it may be lower. For example, if we have a look on the oviduct of the chicken, so one particular gene it is present in superabundant form, and it is albumin, which represents the white portion of the egg protein. So it is present as superabundant. So if it is present in superabundant form, then its direct cloning it may not be required because it is already enriched. When it is already enriched, then it can be directly cloned into a suitable vector, and here the vector that is most preferable, it is M13 MP8 vector. Hopefully you have the idea from our previous lecture that what are M13 MP8 vector and how a screening strategy it is there. So we can clone directly those messenger RNA population that are abundant. It depends upon specific tissue type, as I told you that oviduct of chicken. So after cloning into a vector, then it can be converted into cDNA and then it can be sequenced directly. And after sequencing, then we can compare the sequence to detect the respective protein that it may clone. So in this way we can have a direct comparison. This table, it showed abundance classes of typical messenger RNA population from two tissues. So first tissue, it is mouse liver cytoplasm. It represents polyadenylated population of messenger RNA, and second population it is thick oviduct polysomal polyadenylated messenger RNA. So first column, it indicates the number of different messenger RNA. So first class, it contains 9 different type of messenger RNA, next one 700 and 11,500 in the last class. And next table, it indicates the abundance of molecules per cell. Like first class, that contain 9 different type of messenger RNA, their abundance per cell it may be 12,000, so it is highly abundant. And for the last one, it is 15. So 15 molecules per cell, they are appropriate for constructing cDNA library. And the next one, chicken oviduct polysomal, it contains first class that contain one type of messenger RNA, and it is superabundant. It contains thousands of molecules per cell, and they are very, very appropriate for direct cloning by using phage vectors. On the other hand, if messenger RNA population it is low or moderate, then its cloning it is required in suitable vectors, and the most suitable vectors are the lambda phages. And here two vectors, they are the most important one, like the lambda GT10 and lambda GT11 vectors. Lambda GT10 vector along with lambda GT11 vectors, they can clone around 7 kilobase DNA fragment, and they are the type of insertional vectors. So within the equivalent site, the cDNA it can be inserted, and after insertion for lambda GT10, the plaques can be cloned by hybridization by using specific probe if the sequence of our gene it is known. On the other hand, for lambda GT11, we can insert the our cDNA at appropriate orientation within the lambda Z gene, so that it can be cloned as a fusion protein. And this fusion protein, now it can be detected by using immunological technique. In addition to that, other vectors like lambda ZAP, which is a type of phasement vector, they can also be used for cDNA cloning.